We are going to talk today about genomics to reduce financial risk in dairies, and this video is a combined effort with Amber Adams-Progar at Washington State University, Joe Dalton at the University of Idaho, myself, Holly Nybergs, and Shannon Nybergs. This project was made possible through the generous funding provided by the Western Sustainable Agricultural Research and Education Project, number SW21925. What we're going to cover today includes what is DNA selection. We'll talk a little bit about how we can use genomic selection for mating, costs and reports that were provided by using genomic selection, profitability, when to genotype, who to genotype, and then we're going to conclude with some examples and let you try your hand at genomic selection. So we're going to start out with DNA. DNA is found in almost all of the cells of our body. So here we have a calf, and we know that if we take cells from the skin of the calf, we can see here that it has different cells here at different stages of replication. So we have cells shown here, and inside the cell is the nucleus, that's this round thing, and then inside there is the nucleolus. And we can see that this cell is replicating, and it is got two nucleoluses there, and so it's in the process of making a new cell. And here we have the chromosomes, which are the actual DNA molecules lined up in the middle of the cell, and they have replicated, and they're going to now form two separate cells. So we know that the cell then contains those long molecules of DNA called chromosomes, and those chromosomes, when we open them up, because they're all tightly wound up and condensed, we see that they have basically 4 letters within the DNA alphabet, and those are adenine, which always pairs with thymine, and guanine, that always pairs with cytosine. And this alphabet then codes for proteins that we use in the cell to have lots of different functions in the body. And the proteins themselves, so this yellow beaded structure is a protein, and each one of these little beads then is an amino acid that's coded for by 3 of these nucleotides in the DNA. So when we talk about the DNA, we saw that there were different nucleotides in those, and those nucleotides can vary from individual to individual. And when they vary at a single nucleotide or single spot, that's called a single nucleotide polymorphism, or a SNP. And a SNP isn't necessarily a good or bad thing. It just means that between different animals, they have different variants or nucleotides at that spot in their DNA. So a single nucleotide polymorphism, or a SNP, is a mutation or change in the nucleotide sequence or the nucleotide itself. And single DNA mutations, or SNPs, are what are used when we use genomic selection. And those are the things that we use to predict the performance of an animal. We get SNPs from both of our parents, and the combination of the DNA from our mother and our father, or in this case from the dam and from the sire, is what forms our genotypes. And as I mentioned previously, adenine, in this case shown by the pink bar, always pairs with thymine in this double-stranded DNA structure. You also can see that cytosine, in this picture kind of a green color, always pairs with guanine. And so when we look at the DNA inherited from the parent on the genotype, we'll only get one of these strands from each of the parents that goes to the offspring. So when we look at the DNA or the genotype from an individual, we will look at what the DNA is at one specific location. And so in this one, we see that there's a G here, because you can see that it's purple, and a G also on this strand. All right, so in this situation, that genotype would be called a GG. If instead of looking at this strand, we were looking at the complementary strand of the DNA, it would have been called a CC. Now, because the genotypes have the same SNP at both locations, we call that a homozygote. When we have different SNPs at the same location from the DNA inherited from the dam and the sire, in this situation we have a T here and C over here, so that genotype would be a TC. Or if we looked at the alternative complementary strand, we would see that this would be an A and a G. When the SNPs are different at that specific location, we call that a heterozygote. In this situation, we can see that again it's the same, and so what is that called? It's called a homozygote. All right, now that we've gotten a basic feeling for what DNA is and how that might be inherited, we're going to talk briefly about selection. Selection is when we decide which animals will be in the breeding herd. So we're trying to identify the animals that we want to remain in the herd and produce the next generation of offspring. And so how do we decide that? Well, sometimes we might visually assess the animal, and sometimes this is called phenotypic selection. We might also look at the pedigree of the animal to see if they have outstanding parents in their background. Or we might use predicted transmitting abilities, or PTAs, and we'll talk a little bit more about that in just a moment. And so we can either use our relatives' PTAs or the individual's itself's PTA to make these decisions. Or sometimes we'll use a combination of those things. In phenotypic selection, now remember this is when we are looking visually at the animal to assess it. This is particularly good for traits with little environmental influence. And what that means is that the genetics has the biggest role or say in what that phenotype or the trait that we're looking for is. And so traits that are particularly good for phenotypic selection because they have high heritability, which means they have little environmental influence, would include mature size, or how tall or the stature of an animal, as well as conformation. A little bit earlier, we mentioned predicted transmitting abilities, or PTAs, and PTAs are an estimate of how good an animal is or how their genetic merit is. And so the PTA measures the expected difference between the animal's offspring and all other offspring within the same herd. So here's an example. If we were measuring a trait, in this instance of milking or milk yield, and we had a mean of the herd's offspring was 25,000 pounds of milk, and the average of the cow's offspring that we're looking at was 27,000 pounds of milk, the PTA would be the difference between the average and what her average offspring are, which is 2,000 pounds. So the PTA in this case for milk yield for this individual would be 2,000 pounds. Oftentimes, when we are selecting animals, we will want to select for more than one trait. And so we'll oftentimes use selection indexes for that purpose. We can use predicted transmitting abilities of many traits summed into a selection index, and these individual traits are then weighted either by our priority or by the economic value of those traits to give us a single number from which we can select an individual. When we look at pedigree, or our relatives of that animal's PTA, or predicted transmitting ability, sometimes people will just focus on the sire. And if we focus on the sire, that is oftentimes easy because that information is usually provided by the AI company that has information on the sires that we choose. And so that sire PTA is based on the sire's performance and the offspring's performance and the genotypes that that sire has. Sometimes instead of using just the sire information, we'll use the sire information and the maternal grandsire information and combine those in usually a 2/3, 1/3 weighting. If we have the dam information, we could also use the sire and dam information. So this all sounds great, but there is a slight problem here, and that is that we're not sure exactly what genetic information was transmitted from the parent to the calf, whether we have the sire and dam information, the sire and maternal grandsire information, or just the sire information. And we're going to explain why I say that in the next couple slides. Let's go through how the sire and dam can pass on their genetic information to their offspring. So here we have bull, and he has two genotypes listed. One is a T and an A, and that would be heterozygote. And then we have C and C, and so he's homozygous for C on the second SNP. Here we have a cow, and so she's going to be the dam of the two calves, and she is TT on top, so she's a homozygous for T, and then she has a G and a C on the second set of SNPs. And these are their two full sibling calves. So on the first calf, we see that the sire contributed this chromosome, the A and the C, and the calf got the T and the C from its mother. The second calf got the T and the C from the sire, which is the opposite of what was given to the first calf, and they got the T and the G from the dam, which is also opposite of the chromosome that it could have gotten from its full sibling. When we look at a half sibling, so that means they share a common mother or father, and in the dairy industry it's mostly they share the father. So on this cow, she has an A and a G on both chromosomes. So let's see what the genetic composition that was received both from the dam and sire from the half sibs of these two calves. So the half sib with calf 2 and 3 got the A and the G from the dam, and the A and the C from the sire. So we can see that on this calf number 1, it shares the same chromosome that this calf number 2 had that it got from the sire. So this calf has 50% of the same information for this chromosome from the sire, and it shares none of the same information that we got from the full sib number 3. So if we average this one's a half and this one's 0. When you average a half plus 0 divided by 2, you get 25%. So on average, half siblings share 25% of their DNA. Even though these full siblings don't share any of their DNA for this chromosome because they got the opposite chromosomes, on average, if you look at enough of the offspring, they would share 50% of their information. But you never know which 50%, and that is what is tricky about looking at pedigrees without having the genomic information or the actual genotypes, is you don't know what chromosome they actually received and how much of the information is actually shared between brothers and sisters. Here's another way of looking at it. When we look at relatives for predictions, we're going to have two sets of grandparents here. So here we have a cow and a bull, A and B, and here we have another cow, C, and a bull, D. All right, so as we just explained, we would expect that half of the information from A is transmitted to this bull, and half of the information from B is transmitted to this bull. Similarly, we would see that half of the information is transferred from the dam to this cow, and half of the sire's information is transferred as well. So when E and F are bred, then when we breed E and F, their daughter, G, will have half of the genetic information from E and half of the genetic information for F. So in combination, they would have a quarter of A, a quarter of B, a quarter of C, and a quarter of D collectively from the grandparents. That's what we would expect on average. But let's look and see what could happen. Here we have the same situation, and we have color-coded the chromosomes to try to make it easier to follow. So here we have A has gray and brown, here we have plum and gray or blue, and here we have yellow and blue and purple and green. So in addition to the sire or dam being able to provide one of the chromosomes, in genetics, the chromosomes have to recombine or cross over. That means they have to exchange some of the information with their neighbor chromosome, or the meiotic process that accounts for this doesn't work very well. So we always have to have some recombination or sharing of information between these two chromosomes for the offspring to be viable. So here we see the chromosome that they gave this particular bull was crossed over here at the very bottom. So we have mostly gray and a little bit of brown here. And with the bull, the plum, and this kind of light blue, it had about equal amounts of both of those chromosomes when it crossed over. So here you can see that. When we go over to the other side of the family, we can see that the crossing over occurred at the top here of the yellow one and kind of in the middle between the green and the purple. All right, so now when we get to this generation, we're going to have crossing over again. So not going to just be the blue and the yellow, we're going to have either some green or purple in here. And what we found is that the crossing over occurred here, and so we have green, and we have the green here, and then it went to the yellow, and that's what that offspring got. And over here we had crossing over here at the bottom, so we had most of this gray and then it crossed over to the blue, and so that's what we got there. So when we go back and look at the original grandparents, in theory we should have a quarter of the DNA from each of the grandparents in equal proportion in this heifer. But what we actually are seeing is that though we do have some of B in this particular heifer, it's very small. It's not 25%. It's a very small percentage because that was the result of where recombination occurred and so forth. So we need to remember is when we're predicting the outcomes or the performance of our animals, how much of a particular chromosome or information that they receive from the parent is not apparent without having the genotyping. And so that is the thing that we need to remember is though on average, if we have enough offspring, we would expect to see a quarter of all that information from the grandparents to be equally distributed in the offspring. It may not be the case for an individual animal. The other way we can look for selection decisions is to look at the PTA or predicted transmitting ability of an individual. And so we actually use the genotypes of that animal to determine what we're going to predict their performance is going to be. And this is an advantage because we know more about what was actually inherited from them parents. And this increases our accuracy of prediction, and it can actually make it so that our prediction is equivalent to having about 26 to almost 145 offspring from those individuals because we're able to track the DNA segments that are actually going into the offspring. Because we're more accurate in our prediction, this reduces our financial risk because as we can see, if we choose the wrong animals, and particularly the very poorest animals by a mistake, then we can have severe financial implications because it can cost us a lot of money over the long term, not only in that individual animal's performance, but if she's remaining in the breeding herd, her offspring as well will do poorly, and that will compound the negative profitability situation for our dairy herd. So why are we so concerned with the effective accuracy? Well, by identifying those animals that have low merit, here shown in the gray, we can move our herd towards the genetic progress that we're seeking, which is towards the right. And you might say, well, that'll be obvious because we'll see those animals doing poorly, but we also know that the environment plays a role. And so it could be that these low genetically merited animals may perform okay because they have a better than average environment in a particular situation. And so we might keep them in the herd, and though that they're doing okay, their offspring will likely not do okay. So it's good for us to be able to identify those lower genetic merit individuals so that we can move our genetic progress forward and to the right. Now we're going to talk about some mating decisions. So some people will use genomic selection for very limited purposes. It may be that they're using it just to choose which replacement animals to keep. Or it may be that they use it just to identify or confirm the identity of the parents of that individual. Or you could use it for mating decisions on deciding which animals will be bred to beef bulls or elite bulls, or to be used for embryo transfer or recips, for example. I would suggest that if you're paying the money for genomic selection, that you try to maximize the value or returns that you're getting out of spending that money, and so that you use the genomic selection as a tool for all of those things. So I would use genomic selection for identifying the sire and dam of the heifer, and we know that there's oftentimes mistakes made there on mispaternity. Identify relatedness of heifers and cows with potential mates. You could identify if they have DNA regions that are in common, and so this might help you limit inbreeding. Identify sires that would be complementary to the strengths of the heifer for production and health traits, so that really moves your genetic progress more rapidly. Or, as I mentioned earlier, determine the type of breeding or sire selection that you would like to use. So now that we've gone over some of the basics, let's talk about some of the test costs and reports. The testing ranges for genomic selection depending on the company and what all information you would like to receive, from just identifying parents, from having a broad range of information on health and production traits. The cost ranges from about $15 to about $40, and you need to contact your genomic company to get more details and more specific information on pricing, as that changes from time to time. Here is an example of a report that you might see on some key traits. This is just to kind of give you an idea of what kind of information you would get back. So you would have the identification number of the animal that you tested, and then you would have selection indexes. Here's grazing merit dollars, net merit dollars, and then the rank of net merit dollars nationally. So a number one would mean that they're ranked in the top 1 percentile for that selection index in the country. And then here's some typical traits that you might see, and there's many more here than what we're showing, but we have milk yield, fat in pounds, protein in pounds, somatic cell score, productive life, daughter pregnancy rate, and so forth. Each of the different genomic companies might have a little different acronym here, but they're all defined within the report. Why does genomic selection increase profitability? Genomic selection increases profitability because it improves the accuracy of our prediction of how those animals will do in the future. This allows us to remove those that are more likely to have a poor performance, particularly the bottom 25%. It also allows us to make informed mating decisions so we can increase genetic progress and performance of the herd. And as we do that, and as we have more profitable offspring, we can increase our long-term profitability. This will also allow us to reduce financial risk in the short term, because we will not be having those animals that are performing very poorly, and it'll allow us to reduce financial risk in the long term because their offspring will also do well. So the next thing to consider is, when do we need to genotype? Really, when we need to genotype depends on our breeding objectives. What are your goals for your dairy? Are there particular things that after looking at your records you have identified that need to be improved? And when do you want to sort your females? Because the genomics selection should correspond to the time of when you want to sort your females. If you sort your females from those that you're going to keep and those you're going to cull at a very early stage, you would probably want to do your genotyping early. If you do not sort your animals very early, you might want to genotype later and identify those animals that are more disease resistant in your management system. So I think it's a good time to reflect on what our goals and priorities are for your herd. Do you want to identify replacements? Do you want to identify females that won't produce replacements but that will be in the milking herd? If that is the case, do you want to stratify those individuals and breed some of them to beef bulls or to less expensive bulls? On the other side of the spectrum, would you like to identify elite females and use sex semen with those females, or use assisted reproductive technologies like embryo transfers, or breed those elite females to better bulls? We might genotype early if we identify replacements and elite females and sort them accordingly. If you're going to manage those individuals differently at an early age, this might be a time that you would want to genotype early. This will also facilitate reducing the generation interval by using assisted reproductive technologies. The generation interval is the average age of the cow when her offspring are born. And so as you shorten that generation interval, you typically will make greater genetic progress as that interval is reduced. We might genotype later after weaning or commingling because this would allow us to identify disease risk in our individuals that we're considering as replacements. So if you stratify your animals on those that will be kept or culled at a later time, or if you would like to genotype only those animals that haven't become diseased, then genotyping later may be for you. So who do we genotype? I think one of the most common mistakes is that we decide that we want to keep a certain number of animals and we only genotype those animals. But that doesn't allow you to harness the benefit and the power of genomic selection. You need to genotype more animals than you plan to keep so that you can benefit the most from genomic selection. How many more you want to genotype is something that you should discuss with your genomics company so that you can maximize the benefit of the genomic selection for your herd. So in summary, equal amounts of DNA are inherited from each parent to the offspring, but we're not sure what DNA exactly they're going to get because they have the option of getting one chromosome or parts of one chromosome out of two. Selection decides who will remain in the breeding herd. Genomic selection uses genotypes of the individual to choose who to keep in the breeding herd. Genomic selection improves reliability or accuracy to make good selection decisions, which increases genetic progress. Genomic information should also be used to inform mating and management decisions, and it can reduce financial risk in the short and the long term and increase your profitability. When genotyping is done and what cattle are genotyped should reflect the goals of the dairy. And so this is a time when identifying where your strengths and weaknesses are in your dairy should be used in conjunction with genomic selection to maximize the benefit of this particular technology. So let's now move to an example. And we're just going to use fluid milk as a selection example here, and we'll just go through 5 animals. And then we'll also provide you with another data set that you can work with on your own. So remember, we talked about 3 different types of assessment. The first one was visual assessment. So here we have heifer number 1. and you can see her and see what you think about her physical assessment. Then we can look at her pedigree information on the sire. She had a sire that had a PTA of 422. And remember, a 422 would mean that he has 422 pounds of milk on average for his offspring above the herd mean. When we look at the sire and maternal grandsire information, which is weighting this with 2/3 of the sire's information, which is 422, plus 1/3 of the maternal grandsire information, we see that that number is 345, which means the maternal grandsire must not have been quite as outstanding as the sire. When we look at the heifer information for fluid milk, and we gave you a little bit more of the information like we showed earlier. So this is heifer number 1, and we focus here on the fluid milk. We see that her actual genotypic information predicts that she would have a PTA for milk of 805, which is quite a bit higher than both the sire milk and the sire and maternal grandsire index. So it gives us a little different picture of heifer number 1 than we would have gotten just from her pedigree information, or perhaps even from her visual assessment. So let's go to heifer number 2. Here's a picture of heifer number 2. When we look at her sire milk, we see that it is actually a negative value. So the offspring for this sire on average is lower than the average offspring for the herd. When we combine the sire and maternal grandsire information, it now comes into the positive area, and it's at 251. When we look at her information alone, we see that she's back in the negative at minus 482. So she didn't get the best of her sire or her maternal grandsire when she had the chromosome segregating, and so we would expect that she would have poorer than average performance in milk yield. So here are 3 additional heifers: heifer number 3, heifer number 4, and heifer number 5 visually. So take a moment and see which one you think is best. And then we're going to move on to the next sets of information. The first one is the fluid milk's relatives. So we've gone through heifer 1 and 2. Now we'll look at heifer number 3, 4, and 5, and we see that heifer number 3, her sire milk is very high at 2252 and is the highest of the group. And heifer number 5 is at 1253 is the second highest. Heifer number 4 is third. When we go over to the sire maternal grandsire combination, heifer number 3 still looking outstanding here, with now a change in order of number 4 and number 5, where number 4 now is looking a little more promising than number 5. So now we're going to move on and get the information from the heifers themselves, from the heifers' genotypes or genomic selection. Here's the individual heifer's PTA genomic selection example. So here's the 5 heifers, and remember, we're going to focus here on milk. Here was heifer number 1 at 805, heifer number 2 at minus 482, so she's expected to do poorer than average. Heifer number 3 is 1577. Heifer number 4 is minus 795, so her prediction is by far the worst of the 5. And then heifer number 5 is 383. So when we look at the individual's heifer's information, we get a little different picture than what we had previously. So the heifer that's shining now is heifer number 3, followed by heifer number 1. So there's number 3, our first place heifer when we look at the genomic selection. And heifer number 1 comes in on second with 805 pounds. So just as a reminder, heifer number 4 was second when we looked at just the pedigree information, and now she's worst. Heifer number 3 was the highest milk PTA, but her net merit dollars over here is lower than heifer number 1. Why do you think that is? The answer to that is remember when we talked about the selection indexes, they combine several traits together. And net merit dollars is combining many traits together, one of which is milk. But milk has a low ranking as far as the profitability or the relative weight that is given in net merit dollars. So even though this animal has a very high milk yield, she does a little bit worse in the net merit dollars than number 2, and that is because her reproductive performance is predicted to be worse than heifer number 1. So that was our example for fluid milk on genomic selection. Now let's talk briefly about mating. So here we have bull number 1, and his milk value is plus 1,084. So he has a very high milk yield value here. And you can also see that there are other traits listed here as well: production traits, health and fertility traits, and calving traits. And it also has the reliability or the accuracy listed over here. So his accuracies or reliabilities aren't extremely high, so he's probably a young bull. When we look at this other bull, his values for milk yield is 1612. And you can also see the rest of his production information, his health, and calving traits as well. Also, the reliabilities here are fairly low, so he's also probably a young bull. So when we look at heifer number 1, her strength is her fluid milk, which her value is 805. Her worst trait, when we looked at the different traits on our report, was her fat. Her fat was minus 0.06. When we looked at sire number 2, his strength was milk as well, 1612, and his heifer conception rate was his weakness. And so if we mated her with this particular sire, we've got both of their strengths were the same. So in heifer number 1, she is strong in fluid milk, and we mated her to a sire that was strong in fluid milk, and we call that positive assortative mating, and that is going to drive your genetic progress strongly towards increasing fluid milk. Now the second heifer is a little bit different. Her strength was in reproduction. Her heifer conception rate was 0.9, so she is better than average. And then we have her weakness was fluid milk at minus 482. Now sire number 2 could correct her bad fluid milk at minus 482 because that was his strength. And his weakness was heifer conception rate at minus 2.2. So when we try to take a weakness in one trait, fluid milk for the heifer, which is the strength for the sire, and the strength for the heifer was heifer conception rate, whereas that was the weakness for the sire. When we try to balance those, we call that negative assortative or complementary mating. And so in heifer 2, that's exactly what we did. We mated her to one that would improve her milk yield, which she was poor in, and to a sire that was poor in heifer conception rate because that was her strength. This will drive the genetic progress towards an intermediate value, which is particularly desirable in traits with calving ease and things like that, where we don't want to have a super high birth weight or a super low birth weight, because that middle birth weight is what is going to give us the most healthy calf and less dystocia. Now that we've gone through an example of mating your heifers and genomic selection, please see the attachment where you can click on that, and there is a group of 20 heifers and 2 bulls, and you can go through and make a decision on which heifers you would like to keep as replacements and who you would mate them to. I also encourage you to look at the differences in the net merit dollars or the profitability between the top 4 or 5 heifers and the bottom 4 or 5 heifers, and see just how much difference you would expect those heifers to make over their lifetime. And then consider the cost of genomic testing at being between $15 and $40, and how you would make more than your costs for genomic selection by making wise decisions on choosing your replacements. Thank you for your attention and joining us for this video on genomic selection.